So that means I can talk very slowly.
We'd rather not, no.
Yeah. I might have to apologise. I've just got over a 2-week chest infection. Haven't had my voice for 2 weeks, so I might have to cough and drink quite a lot, but I'll stumble through. This project is a collaboration with InBio Limited, which is a company in Cambridge, UK. I'm based in Melbourne. We spend an awful lot on postage, sending samples backwards and forwards. They have a technology based on heat shock protein complex vaccines. And you'll be stunned to know that at the core of this technology are heat shock proteins. And these intracellular molecular chaperones have a key feature, which is their ability to bind polypeptides. And with that nature, they carry a large protein cargo. These have multiple functions. One of the key things in that is when the cells are stressed, they increase the expression of the heat shock proteins, and stressed cells can have up to 15% of the total cell protein can be heat shock proteins. There are a range of different families of heat shock proteins. I'm not going to list them all. I'm not going to discuss them much. But these are some of the key ones. Ones that we're coming to later include the HSP60 and HSP70. We've heard a little bit about HSP70 before. They have different properties and functions. Some of the really important features of heat shock proteins with regard to my talk today is their ability to interact with the immune system. In fact, they've been proposed that they might act as a link between the innate and the acquired immune system. So they do have a range of functions. They can activate immune cells, and by activating immune cells, they can stimulate the secretion of cytokines and thereby drive inflammation and other responses. They can induce the secretion of chemokines and thereby recruit other immune cells to site of infection or tissue damage. They can also deliver peptides to antigen-presenting cells and be involved in CTLs. And there's been quite a bit of work done on heat shock protein complexes as cancer vaccines, for example. It was also being mentioned yesterday that heat shock proteins are recognized as DAMPs. So in other words, when a tissue is damaged, the heat shock proteins, which are normally intracellular, can be released to outside the cell where they're not normally present, and there they can interact with innate cell molecules. Excuse me a minute. Should have heard me a week ago. I've given 2 talks already with absolutely no voice, so this is a vast improvement. So they are very potent at stimulating the innate immune response when they are located in the wrong position. And this is largely thought to be via interaction with toll-like receptors, in particular cell surface toll-like receptors such as TLR2 and TLR4. And these processes occur via an adaptor protein, MyD88, which signals into the cell. So what I'm talking about is heat shock protein complex vaccines. So this is the technology that InBio works on, and the core principle of this is if you enrich the heat shock proteins from a cell, you can also pull out with those heat shock proteins the cargo that it carries, the proteins. And if you use that as your vaccine, and if you do that from a bacterial pathogen, the heat shock proteins itself can be used to stimulate the immune system against and induce an immune response against the protein cargo that the heat shock protein carry. And with this approach, you then do not require the addition of an exogenous adjuvant. So I'm going to present some work from a couple of the projects we've been working on. These are more models, very early stage work, just really starting to explore the potential of this technology. So the first is something I've been working on way too long in my life, which is H. pylori. So H. pylori is a very beautiful bacterium. It's a bit curved. It's got a bank of flagella at the end. It powers incredibly fast through mucus, and it infects for many, many decades. And it's a definite cause of peptic ulcer disease. It causes 2 types of stomach cancer, multiple lymphoma. And the reason that people have tried for a very long time to make a vaccine against this is its ability to cause gastric adenocarcinoma. There's been, a lot of attempts, and there's been a large number of protective antigens that have been identified. I've listed a few here. There's also been a large number of protective adjuvants with a range of different routes of delivery. And the reason I keep doing this, it's not a twitch, we do not have an effective vaccine against pylori for a number of key major hurdles. And the most important of these is the inability to produce sterilizing immunity. So if you vaccinate mice, this is one of our pieces of data over here. This is the colonization of bacteria. You vaccinate with, say, formalin fixed H. pylori, you can reduce the colonization but you can't get rid of it. And when you translate that to the clinic, it gets even worse, and really there's very little evidence of any efficacy in humans. We've also heard quite a lot of this meeting about the effect that vaccinations, in particular with adjuvant, has on inflammation. Now, the diseases caused by Helicobacter are inflammation-driven. And there's been this phenomenon reported with H. pylori vaccination in animal models that when you vaccinate and then challenge, you actually make the inflammation worse. And that would not be a good thing for a Helicobacter infection. It's this thing termed post-immunization gastritis. You can exacerbate the gastritis. So that's a concern. And at the moment, being a mucosal pathogen, potentially the need for a mucosal adjuvant is not clear at the moment what adjuvant you would use in the case of a human vaccine. Excuse me. So InBio made the heteroprotein complexes, and we've been analysing them. This is our proteomic analysis of the vaccine that they sent to us. And as you'll see, these are just the top 10. This is a mass spec analysis. The first two worked very nicely. You've got the heteroprotein 60 and 70, as you'd expect for something like this approach, are the heteroprotein 60s. So almost a third of the proteins in this is HSP 60, and the next one is HSP 70. The next two down from there, they're in red because these are ones that are already published in the literature as known protective antigens. Then it goes on, and I could bore you for hours talking about the proteomics on this, but I won't. So we started with a simple experiment, subcutaneous vaccination. If you, I say simple because injecting antigens from Helicobacter into mice is a fairly simple way to produce protection. You can just formally fix H. pylori, don't add an adjuvant, and you'll get protection. So this is our first simple experiment. We used the H. pylori HSPC. This was our positive control. And then we added an adjuvant. We used Iscanova, which is effectively Iscamatrix, a saponin-based adjuvant, just to see whether the addition of an adjuvant, A, would this have an effect? And if it did, would the addition of an adjuvant improve it? In the H. pylori model, we vaccinate twice, challenged with the live bacteria, and then later analysed to see what the effect is on a range of different parameters. So this is the antibody response. I'm only going to show serum. I apologise to mucosal immunologists in the audience who are waiting for the mucosal response. Sorry, Alan. This is the serum antibody response. And so this is the group of interest to look at. This is the HSPC. This is post-vaccination, pre-challenge. And vaccinating with the complex without an adjuvant does produce a good antibody response. It's IgG1 and IgG2C. It's pretty much comparable with the other two groups. And this is post-challenge, so after you've actually put the infection as well, and it's a decent antibody response. And this is the bacterial colonization data, so this is the protection. So here's the sham group. This is the level of colonization in the normal mice which aren't vaccinated. This is the positive control level of protection. You vaccinate with the complex, you get the same level. Add the adjuvant, makes no difference. So it is protective, but it's not really exciting. You know, we're really obviously looking for this to come way down before you start getting very interested. We then moved on to mucosal vaccination. We did this via the nasal route. I call it mucosal because when you go nasal in a mouse, you're going nasal, lung, gut. It goes kind of everywhere in a mouse, intramouse. But it's effectively, it's a good route for mucosal vaccination. The dogma at the moment is if you want to mucosally vaccinate against Helicobacter, you have to use a good mucosal adjuvant. So a typical one is cholera toxin, or LT is quite commonly used as well. So it's basically the same experiment, only we're now going nasally and using cholera toxin as the adjuvant. And there's the antibody response. Again, it's a reasonable antibody response, comparable with the other vaccine groups. This is post-challenge. For some reason it was good IgG1, wasn't so good here, but on the whole it was a comparable antibody response and comparable protection. So here is protection with HSPC in the absence of adjuvant, and if you add cholera toxin you don't get any better protection than without it. So it's clearly working in the absence of adjuvant, but it's not making it any better. So that was nice, nothing too exciting. But what was kind of interesting, what we got next when we continued the analysis, I mentioned before the post-immunization gastritis, this observation that you get more severe inflammation. And that's shown here. These are two readouts of inflammation, the mucous metaplasia and atrophy. And if you focus on these two groups with cholera toxin, which are the other vaccine groups. And you've got the moderate and severe. So these two groups are demonstrating the post-immunization gastritis. These mice are infected, they got no pathology. These mice are vaccinated with protection, and they've got moderate to severe pathology. The HSPC had the same level of protection, but they've really got none or mild. So they've got a reduced level of gastritis, which is quite unusual in the system. You normally get that increased severity of gastritis. So that was quite unusual. So we went on to have a look at the gastric cytokines. So these are cytokines present in the gastric tissue. You homogenize the stomach and do ELISAs on the tissues. And focusing on HSPC, you had the highest levels of the Th17 cytokines, 17A and F, had the highest levels of interferon gamma, and it was the only one that produced a significant level of MIP2, which is a mouse equivalent of IL-8. So here you have vaccination with this group that's produced protection, equivalent to the other groups, has less gastritis but has more pro-inflammatory cytokines. And the reason it seems to be doing that is when you look at the immunoregulatory cytokines, what it's also doing is producing IL-10 and IL-13. So the absence of the cholera toxin is allowing this, apparently, to drive a more immunoregulatory response, IL-10 and IL-13. And it's production of these, and actually we know it's IL-10, I've got other data I haven't got time to show you. It correlates more with the IL-10, and IL-10 is known to be perhaps the most powerful immunoregulator in the stomach. So the summary for this is that we've got subcutaneous mucosal vaccination with H. pylori induced a strong humoral response with both IgG1 and 2C. The protection that was induced was as good as the other vaccines, but no better. So it's not something that we get too excited about taking forward necessarily. It produced a mixed Th17 and Th1 response. That's important for something I'll come to a bit later, and this is in vivo. The addition of an exogenous adjuvant did not improve the efficacy, so it seems to be functional by itself, and addition of an adjuvant didn't help. In fact, if you add an adjuvant, what you did was then make the gastritis worse. And this is, say, mice vaccinated with the HSPC only developed that mild gastritis, and that was associated with production of IL-10. So that's the H. pylori part. Now, another model we've been looking at is Neisseria. In particular, we've been looking at nasal colonizing Neisseria. So we've been using these to try and have a look at the mechanism of action for the HSPC. So we've been using two here. We've got Neisseria lactamica is generally recognized as a commensal colonizer of the nasopharynx. And then there's meningitidis, which is one of the words I really hate to try and say. It's one of the major causes of meningitis. There are many different serogroups, and there are 6 main ones that cause disease in humans, which are listed here. And there are now a range of vaccines available against the majority of these. There's 4 of them which are mostly polysaccharide and polysaccharide conjugate vaccines. This type B has recently become available. Whether it's going to be taken up, we'll have to wait and see, which are more protein-based. We're still waiting for an X. And the correlate protection is currently believed to probably be antibody-mediated. But so we're more using this as a model to look at the mechanisms of action of HSPCs. So the dogma, when I started doing this with the company, I said, so how do your vaccines work? And they said, oh, it's tolerant receptors. And I said, what's the evidence? And they said, don't have any. So one of the first things we started to do was have a look to see what is the evidence that it goes via tolerant receptors. So we got in MyD88 knockout mice, because if you look at the tolerant receptors, all of them apart from tolerant receptor 3 require MyD88 signal, and being a DAMP, the HSP is likely to only work when it's released from the cell and therefore activate cell surface tolerant receptors, and is believed, the literature says, TLR2 and TLR4 are the ones that are most important. So really, MyD88 should be essential for its function. So we have done an experiment. We've had wild-type MyD88 knockout mice. We vaccinated them with lactamica, HSPC, and we also used an LPS-reduced form. Because these are gram-negative bacteria, and when you make the HSPC, there's a reasonable level of endotoxin contamination. So another question we wanted to ask is, what's the importance of the endotoxin in the functionality of it? So InBio formulated this, and they went over an endotrap column, and they removed about 99.8% of the LPS. It's down to barely detectable levels of LPS. So we also use this in the vaccine. And we vaccinated the mice. there isn't really a good mouse infection model, so we just vaccinated them and then analysed straight after that. So one thing we did is took the spleens from these animals and stimulated them in vitro with Neisseria lactamica lysate. Excuse me again. And then measured the cytokine response to see what we get. And the first thing that we found was basically the response in vitro was Th1 restricted. So when we stimulated, and this is the media versus lysate, we could get a clear interferon gamma response indicative of Th1, but we got no detectable Th17 or Th2 response. So there was a clear Th1 response, and it was a memory response. So it only occurred in the vaccinated group. We didn't really get it, at least it was much lower. It was not significant in the unvaccinated, so it was a memory response, seemed to be specific to the vaccination. There was also no loss of activity with LPS-reduced HSPC. So there was no difference between this group and that group, and that group and that group. So the LPS, at least whether the residual amount could do this, we don't think so. But there appeared to be no role for the contaminating LPS, and it's clearly MyD88 dependent, because over here in the MyD88 knockouts, we got absolutely no response. So that was interesting. It was Th1, but it wasn't overly surprising because we were expecting it to be a MyD88 dependent response. But then when we looked at the antibody response, then we got something we really didn't expect. Here's the antibody response. This is the HSPC in the wild type, and this is the HSPC in the MyD88 knockout. The antibody response in the MyD88 knockout was completely indistinguishable from the wild type. So the antibody response is MyD88 independent. So whatever the process is for making the antibody response, it does not require MyD88 when you vaccinate the animals. Also, with the LPS, we saw no difference in the cytokine response with the LPS reduced formulation, but we did see a difference in the antibody response. So it was significantly reduced in the wild type. It still produced a response, which is greater than the naive. It was less than the complete formulation. But it is also the same in the MyD88 knockout. So the LPS in the formulation is clearly doing something, but whatever it's doing it via MyD88 independent process. So that's a bit confusing, and that's something we've got to work out. We don't know exactly what's going on. But it's intriguing, and we'll keep somebody busy for a while, I think. We repeated it with meningitidis HSPC. We didn't have an LPS reduced formulation at this time. We effectively saw the same thing. We saw a Th1 response. We saw a MyD88 independent antibody response. We did IgG1 and IgG2C and got a response of both of those. Excuse me. So back to the cellular response. So if the cellular response is MyD88, what is the toll-like receptor that it's activating? And as for the other thing, that also is more complicated than we thought it's going to be. So we started using HEC293 cells. HEC293 cells typically don't express toll-like receptors. They're a human cell line, but they don't classically have the toll-like receptors. And we have these cells here stimulated either with the lactamica HSPC or the LPS reduced formulation, the meningitidis HSPC, or we now have an LPS reduced formulation. And there really is no response as read out by an IL-8 secretion. And these are TLR ligands just as controls. However, if you have a cell line that's transfected with a human TLR2, you can now get a very good response to all four of these formulations. So clearly, these HSPC can activate human TLR2. We don't know about TLR4. So that's fairly straightforward. Okay, we've got TLR2 response. Again, not surprising. When we look onto mouse splenocytes, and we've done quite a bit of work with wild-type TLR2 knockout, TLR4 knockouts, and stimulated splenocytes, say, for 24 hours and looked at cytokine responses. I'm only going to show you a little of the data because it's too confusing for me to get my head around. This is the interferon gamma response. And if you look at the response, this is lactamica HSPC and the meningitidis. I forgot to mention, this is all type B HSPC. And they both stimulate interferon gamma response in the wild type. And they both stimulate a response to TLR2 knockouts, so it's not TLR2 dependent. But when you get to the TLR4 knockouts, while the lactamica will induce interferon gamma, the meningitidis won't. So there's clearly a strain-specific difference in the ability. So what seems to be happening is the lactamic HSPC can probably stimulate both TLR2 and TLR4, and there's redundancy in the system, whereas the meningitidis can only stimulate interferon gamma via TLR4. And this is a T-cell response, and when you go on to, say, a macrophage-based response, such as MIP2, again, the mouse equivalent of IL-8, again, focus on these two. There's no difference in the wild type. There's no difference in the TLR2 knockouts. But once again, there's this difference in the TLR4 knockouts. There is a response, but it's lower here than in the lactamica. So there seems to be multiple mechanisms of inducing it in macrophages, one of which is TLR4 in the meningitidis, but it's not in the lactamica. So there's clear differences on a bacterial level. So just to summarise where I've got up to, there are several mechanisms of actions. Clearly, the cellular response is MyD88 dependent, which is what we expected when we went into this, but there was no empirical evidence for that. I think we now have some empirical evidence. But the surprising thing was that there was a MyD88 independent antibody response, which we've really got to try and work out what's going on with that. The HSPC from both of the organisms induced a Th1, but not a Th2 or Th17 response, at least in vitro. Now, that may be different from in vivo, but certainly in vitro, we could only detect a Th1 response. And there is a clear different mechanism of action between the two organisms. And the HSPC don't activate the same TLRs in the same immune cells. So that seems to be more complicated than we first hoped. So the final summary slide, the HSPC can induce protection or correlates of protection against quite different mucosal bacteria without the addition of exogenous adjuvant and with the induction of good antibody response. But the mechanism of action is clearly quite complex. And it was also potentially variable between bacteria, particularly if you remember going back to the Pylori where we got Th1 and Th17 in that one in vivo. So there were clearly multiple variabilities. So finally, I just want to acknowledge Teng Chon, who did the Helicobacter Pylori work, J.C. Han, who did most of the Neisseria work with a bit of help from Garrett, in Bio, who have been very good collaborators and provided us with the vaccines, and funding from the Australian Research Council and some funding from InBio as well. So thank you very much.
Thank you very much. Question in the front here.
My question is why those vaccination construct together is antibody mediated or cytokine mediated?
It's driven by residual bacteria. That's been shown. So if you have bacteria, because the vaccination doesn't clear the infection, you have some bacteria left. And it's shown many years ago, if you antibiotic treat the infection and get rid of it, you don't get that. So the residual bacteria will drive that. It also seems to be a transient phenomenon. So if you leave it for, say, 3 or 6 months, then it evens out. So there's no evidence for this. What I personally think it is it's an accelerated normal gastritis, because you have a naive animal, and the development of gastritis in the mice is quite slow. If you vaccinate it, you're effectively priming the memory immune response. So when you then go in with the infection, you're now infecting a non-naive animal, and you get an accelerated gastritis. So it's almost like just bringing forward the normal gastritis that develops. That's my personal view. And so if you leave it for 3 to 6 months, eventually the normal gastritis process catches up, then they equalise. But I think it's just accelerated gastritis because you're priming a naive animal. But there's no proof for that.
Well, that's really great. Do you get further clearance with subsequent boosters?
With the Helicobacter?
Yeah.
No.
That's it?
That's it, really. Yeah, unfortunately. We've tried all kinds of... Well, not necessarily. We haven't tried with HSPC. I've tried it with anything else I could possibly think of and lots of other people. I mean, I've been doing H. pylori vaccine experiments for 18 years, I think, which is terrifying. But, yeah, and it's been... and lots of other groups. I think Harry did. Didn't you do Helicobacter work many years ago, Harry? I think. Yeah. I remember seeing your name on lots of papers.
So do you really think, given the data you show around sort of basically decreased inflammation. Do you really think, do you need sterile immunity, I guess, is the question. Would you come along then with an
For a vaccine, yes. And
for an antibiotic therapy on top of that? Would that be an approach?
well, that's nice on an individual basis, but it's not really practical on a population basis. And the countries that really need it is like Japan, China, Russia, and to go in and vaccinate and then antibiotic treat is really not practical at a population level.
I'm just guessing if there's less antibiotic required. I'm just trying to think of some sort of combination. Sorry, keep this microphone. Some sort of combination. And given that's, I think, from all the data I've seen on the Helicobacter immunisation, that plateau seems to occur frequently.
I think the reason you get that plateau, again, personal opinion, is there's a very specific T-cell response that virtually any antigen can work. It's almost remarkable how many antigens can be protective. And you get a specific T-cell response, but there's a non-specific effector mechanism. No one knows what that is yet. And what we need to do is make a vaccine that creates a specific effector mechanism. And that should be an antibody, but these bugs are so clever that they can evade the antibody response. Somehow we have to get around the ability of them to evade the antibodies. So all you need to do is get a T-cell response, and you can get something like that.
And they're also difficult to get to with antibody.
Exactly, yeah.
So what was the source of this? It showed protein 6. What was the source? How did you get it? How did you isolate it?
InBio.
Pardon?
InBio gave it to me. No.
You bought it?
InBio have a technique that they've patented. So it's their technology. It's based on, it's an enrichment process based on IP, largely, I think.
Yeah, but it's
PI,
highly,
sorry, not IP,
this
PI.
H protein is highly conservative among bacteria. Quite conservative, I mean.
Yes, yes, highly conserved.
But, and how do you check it? By proteomics or each time, or how do you do this? Because might be a little bit viable, you know. It's not, if it is homogeneous enough, each time you get the same batch to batch variation, if there is any difference.
Well, the Neisseria ones we're using are made by a company under GMP conditions. Mm-hmm. So they're making, because they're InBio are approaching clinical trial, so they're making their vaccines for clinical trials, so they're GMP conditions. So I'm hoping there's no viral contamination in there.
Okay, thank you.
Good. Well, thank you very much. Harry, one last question.
Just maybe a bit of a comment based on your efficacy data showing the lack of sterilizing immunity. From what I remember with our earlier studies, and we did a whole genome assessment of Helicobacter pylori, and we never ever found an antigen or an antigen cocktail that would give sterilizing immunity at all. But what we did find was that when you combined vaccination with kind of a dual therapy, so an antimicrobial therapy, that you actually could achieve a sterilized stomach from the perspective of the level of bacterial burden that in fact was sustained with immunization as well. So you didn't get a rebound of infection when you combined vaccination with antimicrobial therapy.
But then they weren't protected against re-challenge, though. I think.
I'd have to go back and check. I mean, I can't remember whether they were sterilized, whether we did the re-challenge studies actually. It was, for us, it was at the time we were just looking at whether you could actually get greater than a 2 or 3 log reduction in bacterial burden. But I can't remember. I'd have to go back and check.
Okay, thank you. Let's move to the last presentation before lunch by Zakaria Mom.